This playlist will consist of all the videos regarding blood physiology of MBBS first year. So, here presenting the first chapter, Introduction to Blood and Plasma Proteins. So, what is blood? It mainly consists of plasma and cells present in it. So, here our main focus will be on the plasma proteins and blood. So, here you can see in the plasma there are three main proteins. Other proteins are also there, but mainly albumin, fibrinogen, and globulin are present. So, what's the function of these proteins? They maintain the colloid osmotic pressure. So, why it is called the colloid osmotic pressure? Because when proteins they combine with water, they form a colloidal solution. So, that's why it's known as colloid osmotic pressure. So, these plasma proteins they help to maintain the colloid osmotic pressure. Now, what is the function of these proteins? So, we will study the function one by one. Now, for the albumin, it maintains the osmotic pressure. As we have studied that all the plasma proteins they help to maintain the colloid osmotic pressure. So that's why albumin also helps to maintain the osmotic pressure, but it has the maximum contribution in forming the colloid osmotic pressure. Secondly, it also function as a carrier protein. So what does it carry? It carries steroid, fat, sex hormone binding globulin, corticosteroid binding globulin, and testosterone binding globulin. Means these are also the hormones which are carried by the albumin. So it also act as a carrier protein. Now fibrinogen, they help in coagulation or clotting. Also they are the precursor of fibrin. Means that fibrin is formed from the fibrinogen when acted upon by the thrombin. Now globulin, they help in the defense mechanism. Now, beside albumin, fibrinogen, and globulin, other proteins are also present. So they are helpful in MCQs. So transferrin. So you can easily remind it like trans means transport and ferrin means iron. Means transferrin help in transport of iron. Now haptoglobin. Globin say you can remind that HB hemoglobin. So it transport hemoglobin. Hemopexin helps to transport heme. Now, where all these plasma proteins are formed, mainly they are formed in liver. They are formed as 30 gram per day, and so as we know that they are formed in the liver. So, if there is cirrhosis of liver, then their production will decrease. The albumin, the globulins are produced in the lymphoid organs. Now, a further topic is there Stalin forces. So, these are hydrostatic pressure and osmotic pressure. There are two types of these forces. The hydrostatic pressure is applied by the interstitial fluid and also capillaries, same for the osmotic pressure. So what are these pressure? So imagine these are the capillaries. So the pressure which help to move the substances out from the capillaries is known as the hydrostatic pressure, and osmotic pressure helps to hold the substances in it. So here it is the cell. Here these are the intercellular matrix. They are the extracellular compartment. Extracellular compartment consists of interstitial fluid and capillaries. So interstitial fluid and capillaries they exert the hydrostatic pressure and the osmotic pressure. Now osmotic pressure. So we are telling that the plasma proteins they exert the osmotic pressure. Then how much osmotic pressure they exert? So in total 28 mmHg is the total osmotic pressure which is exerted. Of this, 19 mmHg is exerted by the proteins and 9 mmHg is exerted by the Donen effect. So what is this effect? That beside proteins, further ions are also present in the plasma. So they also exert the pressure, colloid osmotic pressure. So in totality, they form the 28 mmHg colloid pressure. And as I have already told you that albumin has the maximum contribution to it. Another concept is there reversible equilibrium between plasma protein and tissue protein. It simply means that there is an equilibrium between the proteins present in the plasma and proteins present in the tissue. So whenever there is any deficit of proteins, then this is maintained by maintaining the equilibrium. So as I have told you that there is state of equilibrium. What is the significance of this? That when there is any deficiency of proteins, then we can intravenously inject the proteins into blood. Then these will definitely flow towards the tissues, and hence there is maintenance of the equilibrium. Within hours, the proteins will flow. Now another concept, or we could say the last concept of this chapter, is separation of plasma proteins. Sometimes there is need to separate the plasma proteins, mainly albumin. So there is a procedure in which By applying salt and ethanol, there is change in pH and temperature, and this change affect the solubility of the proteins, and then these proteins separate out from the blood. Then we can centrifuge them, and after that by electrophoresis we can easily separate these proteins. So it's known as the separation of plasma protein. You can remind that by applying salt and ethanol, and it will change the temperature and pH, and hence the solubility will also change. By centrifugation and by electrophoresis we can finally separate the proteins. Another topic is plasmapheresis. It's also included in the separation of plasma protein. So plasmapheresis, in this we remove the plasma from the blood. Sometimes there is excess of plasma proteins in the blood which make the blood viscous and hence affect its flow. So there is a need to separate the plasma, but in this procedure we will not separate the RBCs. We will take the blood, we will take out the plasma which contains more proteins or affected plasma. So we remove that plasma and then transfer the blood back to the human. So it will not affect the RBC count, we will just remove the plasma. So when there is a need to remove the plasma, when there is excess of plasma protein which makes the blood viscous, and the second thing is when there are certain antibodies present in the body which affect the body tissues. So here the chapter is completed. Now if you like the video, please like, share, subscribe. Thank you.